This is Dr. Sewanti Limaye. I'm Director of Precision Oncology in Sir H.N. Reliance Hospital, Mumbai, India. I'm here today to talk to Dr. Razelle Kurzrock, who's the Chief Medical Officer at WIN Consortium, the World Innovative Network in Precision Oncology. I'm here today with Dr. Razelle Kurzrock, who is also a professor of medicine at Medical College of Wisconsin and head of Precision Oncology Center at Medical College of Wisconsin. I'm here to talk to her today about many of the areas that are left unvisited in precision oncology. And it's an honor for me to be interviewing her on behalf of Journal of Immunotherapy and Precision Oncology, JIPO. And so, Ray, a very good afternoon to you and welcome to this interview recording. I am thrilled to be sitting across you and talking to you because we've always held you in very, very high esteem. You're one of the pioneers in precision oncology and literally have founded this entire effort. So I have some questions for you, but before that, thank you so much for doing this with us.
Well, thank you, and it's a pleasure to meet you in person. We've, of course, interacted over Zoom many times, and I mutually feel honored to meet you. I think that you are a real world leader in precision oncology, and one of the reasons I was so excited to come here was actually to meet you in person. I feel like I know you well, having spoken to you over Zoom so many times, but it is nice to meet people face to face.
Thank you. Thank you, Ray, and thank you for all the encouragement that precision oncology follower in a developing nation needs, and thank you for the encouragement. So with that, I'll ask you, what is your vision for precision oncology at large, in terms of globalization of this effort?
Yeah, so I think that there's many parts to the vision, but first we have to understand what precision oncology is. And what it is, it's understanding what is wrong with an individual tumor at the genomic, the transcriptomic, the protein level, and then customizing the therapy for that individual. And because tumors are complicated and they're all different from each other, in order to personalize therapy, you have to be very precise. And you have to understand each tumor's molecular makeup. And so that's where the word precision oncology comes from. And so the first thing is to have technologies available so that we can understand in depth the tumor's makeup. And then the second part of that is to understand that this is not just about genomics. This is about genomics and transcriptomics and proteomics and immunomics. And furthermore, it's not just about gene-targeted drugs. It's about immunotherapy, understanding which patients should get which immunotherapy. It's also about hormonal therapy. It's about chemotherapy. You brought up in the WIN Consortium, what about chemotherapy? Chemotherapy are each targeted drugs. They were just developed before we understood the targets. And we all know that chemotherapy works very well in a subset of patients. What we could never figure out is, what is that subset? So I think we have the tools now, or are close to having the tools to understand chemotherapy as well. So it means putting all the types of drugs and all our tools together in order to make patients better. And then the other part of this is democratization. Right. Of this effort, because right now the effort is concentrated in communities that may be wealthier, let's put it that way.
Right.
And where I think this needs to go is that everybody, that testing at the molecular level is done as a reflex for everybody. So for instance, everybody gets a diagnosis, whether they're rich or poor. You put the slides under the microscope and you say you have colon cancer or you have breast cancer. That's true across the globe. Well, now we need to put the slides under the molecular microscope, and that should be true for everybody. It should be just as essential as having it under the light microscope to understand the pathology. It actually should be even more essential. So that's where I think precision oncology is going. I think that's where the future is.
I think that's so well said. The molecular microscope. So we think of routine histopathology as mandatory. We have to have a histopathologic diagnosis for a tumor before we treat it. We characterize it based on that. But the molecular microscope gets left out. When we talk about scaling, bringing it to the masses, you're really actually not testing the masses. And that's your recommendation, statement right now points to that. One should have this as a reflex testing so that each patient has that answer when they're starting therapy.
No, absolutely. It should be reflex testing. And I'll even go even further and say something that may be controversial. I think categorizing patients on the basis of the way they look, the tumor looks under the light microscope, rather than under the molecular microscope is an accident of history. It's because the light microscope was invented 500 years ago, and the molecular microscope was only invented 20 years ago. And it wasn't actually inexpensive enough to do until the last 5 years or so. So we have a tool that's 500 years old, and that's why our reflex is to look at every tumor under the light microscope. But that's an accident of history. The molecular microscope is the much more powerful and the much more relevant microscope. But it's the newer microscope. But we have to get there where it's a reflex testing. And I think the price has finally come down enough that we can go there. Before it was so expensive that we couldn't go there. But now the price is such that I think that it could be reflex testing for every single cancer.
Wonderfully said. I think that's the message that we need to project, convey to the world, that with the molecular knowledge of the tumor, the approach to therapy, and especially, as you said, in the consortium, if we did it at the start of therapy and not when the cancer had already mutated and become a difficult to control disease, that the real application of precision would come in. We understand from a molecular microscope how the disease looks and apply therapy based on that.
Yes, absolutely. And I think, the question was raised during the WIN Consortium about chronic myelogenous leukemia.
Right.
Chronic myelogenous leukemia is absolutely the example. When I was young faculty, it was a death sentence. 100% of patients died. They died in 3 or 4 years, and there was nothing that could change that. Today, patients have a normal life expectancy. But it's because patients are treated right at diagnosis. Nobody waits to end-stage CML to start molecularly matched therapy, which is essentially imatinib and second and third generations of that. The patients are treated right at diagnosis. That's not what we do in solid tumors. We wait until the patients have metastatic disease. And if you wait with chronic myelogenous leukemia till accelerated phase or blast phase, imatinib, which is the miracle drug that gives patients that previously had a death sentence a normal life expectancy, it does not work in accelerated and blast crisis. Essentially, it's almost worthless at that point in time.
And so in solid tumors, you're actually letting the blast crisis type of situation happen.
Absolutely.
Without really intervening right at the start.
Absolutely.
When you understand.
Yeah.
So I really envision and hope for the day where we are applying precision at the start.
right at the start, as soon as the patient is diagnosed, not waiting until they have metastatic disease all over the place. When they have that 1 centimeter tumor, that's when. That's it.
Strike.
Absolutely.
Well said. So, changing gears and talking about drugs, your thoughts on availability of novel drugs outside of the approved indication? Because, you've spoken for drug repurposing. You've also spoken and you have utilized drugs in areas where they may not have a classic approved indication. And I want to project that to the precision crowd.
Right. So I think that this is a very important issue. And I don't know all the answers. And of course, the regulatory issues are different in every country. They're different in the United States. They're different in Europe. They're different in India. I will give you a feel for the environment in the United States. So in the United States, physicians are legally able to prescribe off-label drugs. And historically, if you look at the published statistics, oncologists always used off-label drugs. Probably about 40% of oncology drug use in the United States has been off-label. So it's nothing new in the United States to use cancer drugs in an off-label fashion. What is new in the context of precision oncology is to use them in a smart way rather than randomly giving patients off-label drugs and trying this or that, to try to match the drugs to the molecular alterations in the patient. And so because there's a long history of off-label drug use in the United States in oncology, this smarter off-label drug use is nothing new.
Right.
It's legal, and it's really builds on a history of what we've been doing, but not in a smart way. But there's still a major barrier, and that's for who pays for those drugs. And there's a variety of ways. Sometimes payers actually pay for off-label drug use, and sometimes they don't. And there's, of course, other ways to get off-label drugs. But at some point, I think what we're going to have to address is how do we more officially authorize this drug use? How do we prove that this helps patients? The FDA has been amazingly, I think, had amazingly progressive thinking in the cancer field. Right. With the tumor-agnostic approvals and so forth.
Yes, of course.
And but this issue is going to have to be addressed, especially if we're going to democratize the availability of these drugs to patients who need them. And how do we weigh out against situations where drugs may be prescribed where they're not needed, or the costs escalate? And I do want to mention this cost escalation, because people keep on thinking, well, we're going to use these off-label drugs that it's going to cost more. But if you do precision medicine correctly, it not only tells you which drugs you should use, it tells you which drugs you should not use. And I spent a good deal of my career giving FDA-approved drugs to patients with solid tumors, and it didn't help them. Didn't help them at all. Now that is not only heartbreaking for the patient, but it's a complete waste of money. Resources. And so people keep on thinking, well, how much is this going to cost? But I really want to emphasize that part of this is avoiding drugs that are not worthwhile, that are not going to be useful for the patients. So I think that will at least equal or more the cost savings for not using useless drugs. But the regulatory issues will still need to be worked out. And of course, I address them in the United States. and the regulatory issues are completely different in other countries. For instance, WIN Consortium is international, and one young faculty approached me just this morning from one of the Asian countries, and I'm not going to name it, but she told me that they're not legally allowed to prescribe off-label drugs.
Such a limitation.
That is a real limitation for precision oncology. So she was asking me, so how can we solve this? I actually don't know. If you can't legally prescribe the off-label drugs, there's not a good solution. Maybe a solution would be that the government allows within the context of a clinical trial.
Or a local consortium.
A local consortium, that the drugs be legal to prescribe, providing, let's say there's a molecular tumor board that recommends them, and the patients sign consent and they go on a clinical trial. Maybe that is the solution to that issue.
I think I couldn't agree with you more that you need validation. And in countries where regulations are just being carried out, to move away from guidelines and regulations may be a taboo or illegal. That's where such consortiums and groups of oncologists really are meaningful, to validate that there is such approach where non-formulary, non-indicated or organ-agnostic approach is genuine.
Right.
And I think precision needs that kind of support. In league with that, your thoughts on molecular tumor board. I know you have supported molecular tumor boards in a big way. And in your own trials, you have large representation of molecular tumor board directed therapy.
Right.
So just some light on that.
Right. So I think molecular tumor boards are very important. And there are several features that need to be present in a molecular tumor board, and they're not always present. So one important feature is, just like for any tumor board, it's important that it be multidisciplinary. Right. The other feature is that it's very important that the faculty that head the molecular tumor board be knowledgeable in the area of genomics and molecular matching and drugs. Now, that may seem like obvious, but it's not. I've been at molecular tumor boards where people have picked a young faculty member that may be really smart, but they don't have the experience. So it really is. It's just like you wouldn't have major heart surgery by somebody that just entered their cardiovascular or surgical fellowship. You don't want a molecular tumor board run by somebody that's just dipping their feet in it. It's a very complicated. It's a very complicated field. And then I think it's also a good idea to either have a clinical trial that allows consenting the patients and then collecting the data, or have some sort of means to collect the data on what you're doing. So it's an iterative process so that you learn from it. Now, one of the really amazing things today, not too much good came of COVID, but there is some good things, and that's that you can have a national molecular tumor board, or even an international molecular tumor board. Sometimes in the past, people ask me, well, we don't have the local expertise. What do we do? Well, the world is open to you now. You can have expertise from across the country. You can have expertise from across the world, not just the country. And so that idea, which was really a problem if we go back 5 years ago, we don't have the local expertise to do that. That's just not true anymore. You just have to look at who knows what they're doing, who has the expertise, and bring them in. We have in the WIN Consortium, we had the clinical management committee for the winter trial. This was like a molecular tumor board. Yeah. And we had experts from multiple continents. I'm involved leading a trial for target cancer. It's called the TRACK trial for rare cancers. It's a national trial, but we have experts participating from all across the country. Wow. We have experts from MD Anderson. We have experts from UCSD. Again, right across the country. So I think it's a whole new era, and bringing these experts together is now completely possible, even if you're missing something locally.
I think that is one of the most critical points that help with application of precision. Because the field is evolving. There's so much data, and it is a complicated subject. It may not be possible for one person, really, to be able to define the strategy. It may take a whole group of people. And just as oncology was evolving and we had multidisciplinary care, now that precision oncology is evolving, we need to have participation, and we also need the molecular tumor board efforts to be led by experienced people and get experience from across the country, across the world where there is a lack of. So I think these are extremely important points that lay the basis for application of precision. So a couple of my last questions to you are towards application. And in the application arena, one issue that really stands out is drug sensitivity testing. And your thoughts on in-lab drug sensitivity or patient-derived xenograft testing. And then my last question will be about clinical trials. So regarding the drug sensitivity, do you believe in it? Do you feel it needs to evolve? Where do you think it stands?
So we've had less experience with drug sensitivity testing, but I absolutely believe that is a very important component and has a strong future. The xenograft testing historically has had a problem, and that's the length of time to get the results, because many of these patients need results right away. But what has evolved more recently is functional testing. Yes. That is much quicker, sometimes can be done in days. And I think that the evidence is accumulating that this could be very important. Why do you need that? Many of these patients have complicated pathways. They don't have just one aberration. They may have many aberrations. And what functional testing allows you to do is look at the sum total of those aberrations. It also allows you, we spoke before about chemotherapy. Right. It allows you to look at chemotherapy. These are very important drugs, but allows you to pick chemotherapy. Should you give gemcitabine? Should you give a taxane, and so forth. So it allows you to look at a range of different drugs. So absolutely, I think this has an important future. I think in the United States, in order to use it for prediction, in order to use it to treat, we need it to be clinical grade testing. Yes. And I think everywhere we need the results to be relatively quick. The results that take 2 or 3 months to get, that's not going to work. But the results now in some of the modern assays that could be back in a few days or in a week or two, absolutely very important for the future.
Should be developed, should be utilized.
Yes.
Excellent. And so now coming to one of the most important application questions regarding clinical trials. We've talked about umbrella trials, basket trials, but I've also heard you talk about N-of-1 trials and requirement for novel trial designs, and I predict was one such trial, and I'm sure you are working on many more. So your thoughts regarding where the precision clinical trials should be going?
Right. So very important question. I think that the future is N-of-1 trials. And so let me try to explain what an N-of-1 trial is, because the immediate reaction is, well, how do you get information out of one patient? So let me backtrack a little bit, and for the audience, explain an umbrella trial and a basket trial. And these are definitely move forwards, but the next generation is N-of-1. And this is what we did with the iPredict trials, actually what we did with the Winter trial, and it's what we're doing with the TRACK trial from Target Cancer. So an umbrella trial is, for instance, a trial in lung cancer where we look at multiple different genomic subsets. So we may look at patients with RAS mutations and patients with EGFR mutations, patients with ROS, and patients with ALK alterations. So it's an umbrella over different genomic subsets, and we match the patients to different drugs. A basket trial is kind of the reverse of that. Now we put the genomics front and center, and I think this is a very important move forward, and it's something that Vivek Sabaya talked about a lot during his keynote here at WIN. These are the tumor agnostic trials, where, for instance, we're looking for a RET fusion. Mm-hmm. We don't care if the RET fusion occurs in lung cancer or colon cancer or breast cancer or in some rare tumor. If the patient has a RET fusion, they get treated with a RET inhibitor, and that's a basket trial. So all the basket trials and the umbrella trials, I think, are a real move forward because they're matching patients to drugs. But once we started doing next-generation sequencing on patients, we found what I'll call a fly in the ointment, and that's that the majority of patients don't have one abnormality. Most of them actually have multiple abnormalities, and that means they don't have one driver. And then even to make it worse, what we see is that everybody's abnormalities are unique. They're all different from each other. This is why we call it personalized medicine. Right. So now we have a problem, but it's a problem created because we're used to doing things in a certain way. What the science has revealed is that everybody's tumor is unique, and everybody's tumor is complicated. So what does that tell you? Well, that we need a customized combination of drugs for each patient's tumor. But that's N-of-1, right? And so the problem isn't what the science is telling us. The problem is, how do we break the mold of what we've done for the last 100 years and do something differently? Well, the first answer is that you have to break the mold because the science wins. You know, this is the data. When quantum mechanics was discovered, it was like crazy. It's still hard to understand quantum mechanics, but that's the scientific truth. You have to deal with it. I think the same thing for molecular typing of patients. It revealed that the tumors are complicated. It revealed that they're unique, and now we have to deal with it. Fortunately, there is a way to deal with it. And instead of looking at every combination in every patient or patient type, which is impossible. If there's 300 drugs in oncology, 3 drug combinations is 4.5 million. And then if you're going to look at that in every tumor type, you know, you're going to
just multiply X number of times.
Yeah, you're going to get it into the billions. Those studies are not going to happen. Impossible. But what you can do is you can look at the algorithms that navigate you to certain combinations and see how accurate the algorithms are. To summarize, instead of trying to look at millions of different combinations of drugs in several hundred different types of tumors, which is going to be several billion trials, which cannot happen. We should look at the algorithms and how well does a particular algorithm predict how patients do. So the algorithm will navigate patients to different, each patient will get a different combination. And we're not assessing the combination, we're assessing the algorithm. And that's, the algorithm may be derived in many different ways, whether it's machine learning or other ways, but that's what will get assessed. Eventually, I think that's what's going to get regulatory approval, the actual algorithm. And then the drugs decided on by the algorithm should then be available because we know it's an accurate algorithm, or it's the best algorithm that we have.
Wow. So when I look at this and I hear it from you, it sounds very futuristic, but knowing how precision has evolved, and in your own words, from your own talk, the iceberg and the multi-layers of the iceberg, and the picture from the consortium today where Dr. Subaya also shows that elephant where gender equality was practiced and you had equal number of women and men trying to interpret different parts of the multi-omics in the elephant. I don't think the algorithm will be that futuristic. I think it is going to happen in the near future because world is moving towards that algorithm that you're talking about. So, Dr. Kurzrock, final thoughts for the viewers, the precision oncology followers.
Yeah, so I think my final thoughts, putting it all together, is that the future is in customized combinations of drugs, that all the different types of drugs need to be considered, whether they're gene-targeted drugs, whether they're immunotherapy, chemotherapy, or hormonal therapy, that we need to treat earlier in the course of disease, waiting until patients are metastatic or even worse, treatment refractory metastatic. It's nice that we see responses, but if we want to cure people, we need to be treating them early in the course of disease. And then finally, we need sophisticated algorithms, and we've already showed that this is possible through the iPredict studies that direct patients and use multi-omic and functional analysis to direct patients to certain combinations of drugs. I think just like there's more than one drug to, let's say, target RET, there's going to be more than one algorithm. And we're going to need to evaluate the value of different algorithms.
Amazing. Thank you so much.
Thank you.
Thank you for giving all the time to us, for your viewers in Precision Oncology, for Journal of Immunotherapy and Precision Oncology. And this message that you've given gives us a lot of hope and also helps us be a part of your vision, which I feel is most critical because this is an evolving field and we really need to be guided by world leaders and pioneers in the field like yourself. And from this interview, I hope that all of us will get some very salient messages, and one that stands out for me, actually two. One, early application of precision, and two, customized protocols, N-of-1 strategies. And we'll wait to see much more coming from you, your team, and the WIN Consortium. Thank you.